Rita Carsetti. I am the president-elect of IOIS, and I'm here to welcome you to Vienna for the celebration of the Day of Immunology. All the friends you see behind me will explain to you why we are celebrating this important day in Vienna. The subject here, they are saying welcome to you. So the topic today is neuroimmunology, a very important subject because immunology will explain the mechanism of disease and also develop the tools, has developed actually already tools to cure some of these diseases. So to welcome you, there is a first speech, which is from Miriam Merad, who is the now president of IOS. Welcome, Miriam. The stage is yours.
Okay, so hello everyone. My name is Miriam Merad, and I am a professor of immunology at Mount Sinai School of Medicine in New York. I'm the president of the International Union of Immunology Societies, not for too long, and I'm pleased to celebrate the Day of Immunology 2025 with you all. So first I want to thank Rita Garcetti and Ursula Wittdomen-Schmidt for organizing this exciting day of science and celebration, and indeed there is so much to celebrate as we are living through really a moment of extraordinary momentum in immunology. Thanks to advances in technology and AI, we are unlocking the immune system at a pace we've never seen before, right? And we are discovering therapies that are transforming medicine. And in fact, the biggest blockbuster in medicine today are immunotherapeutics. So this is also just the beginning, right? From cancer to sclerosis to Alzheimer, we are realizing that inflammation is at the root of every major disease. And this year's theme, Neuroimmunology, highlights one of the most exciting frontiers in our field, the immune system's deep connection to the brain and the nervous system. So even as, however, scientific discoveries accelerate, we are also witnessing growing mistrust in our mission. So some view science as too costly, even though research budgets remain disproportionately small compared to other budgets in society. And recently, several areas of immunology research were discontinued at the National Health Institute in the United States without any consultation with the scientific community. So it is important for all of us to remain engaged, and we must remember that an attack on science anywhere is a threat to progress everywhere. So today I urge all immunologists in the world to be united, right, and to fight together in one voice, really against any restriction that prevent, the progression of our field, and especially this extraordinary field of immunology science. And let's make sure that we strengthen our network and really commit to keeping this field vibrant, open, and bold. So I wish you all a fantastic day of immunology. Thank you all for being here, and I'm excited about the day to come. Thank you.
Thank you, Miriam. This was a very inspiring talk. I'm really very grateful. So we have now another introduction that is from the president of EFIS, of the European Federation of Immunology, and it is Bojan Polic. Bojan.
Thank you, Rita. Thank you, Ursula. So on behalf of EFIS, I would like to welcome all immunologists worldwide to this Day of Immunology and to celebration of immunology and to this webinar. I would also like to thank all our European member societies, national societies, and individuals which are celebrating also this Day of Immunology and doing all kinds of activities regarding this day. I'm currently in Istanbul on the MIMBI conference and also celebrating the Day of Immunology with Turkish Immunological Society. So today, this topic is, I'm very happy that we opened this topic, brain and immunity, which is actually a topic which is, has been investigated for a long time, but maybe in the last couple of decades, tremendous progress has been made thanks to a lot of different tools and genetic tools and imaging techniques. And I think now we are actually coming to very mechanistic insights into the interaction between the immune system, how immune system actually causes different diseases and disorders, neurological, and how the immune system can also regulate the immune system and that it can cope with different threats. So today we will have actually several excellent talks, and let's enjoy in these talks and let's enjoy in Day of Immunology. Thank you.
Yeah, thank you, Bojan. Also from my side, a warm welcome from Vienna. My name is Ursula Wiedermann and I'm the chair of the vaccine committee from IOS. We chose to celebrate the Day of Immunology from Vienna because in a few months we will host the 19th International Congress of Immunology here from Vienna, and we would like to inspire you all to come to this beautiful city. More about will follow later, but now we will start with our excellent presentations, and I'm very much looking forward on the first speaker, Sophie Ugolini. She's from the INSERM in Marseille. Just a few introduction words. Sophie has been doing her PhD in immunology in Marseille in France, and she has had 3 times visiting as a visiting scientist abroad. She was at the Scripps Research Institute in La Jolla in California. In 2013, she was at the Rockefeller University in New York, and followed then by the Peter Dorothy Institute of Infection and Immunity in Melbourne. And since 2015, she is the principal investigator of the ERC Consolidator Program, Neural Regulation of Immunity. And her group aims in several years to determine the precise impact of the nervous system on the immune responses, and to understand the underlying mechanism of this regulation and how this interaction shapes the host response to pathogens and injuries. And with this, Sophie, we are very happy to have you online here. Sophie will also be online then for afterwards for questions and answers. And I hope you hear me. Welcome, Sophie. We're looking forward to your talk.
Good afternoon. Thank you very much for the kind introduction and for the invitation. I'm going to share my screen. Are you able to? No, it's not the good one. Sorry.
Broaden.
I think it's the wrong presentation.
It should be on full screen.
No, I think she said it's not the good one. I think she's wrong, the wrong presentation.
It's always complicated.
Yes. Can you see my presentation?
Perfect.
Yes.
Thank you. So I'm really pleased to share with you today our most recent results on neuroimmune pathway orchestrating tissue repair and host defense. And also, I'm happy to celebrate with you our shared science, progress, and impact on health. In 2019, the COVID-19 pandemic revealed how vulnerable humanity and our societies are to emerging infectious diseases, and scientists and physicians were on the front lines. Today, a growing hostility towards science and scientists points to another type of crisis, and a loss, which is a loss of trust in expertise and in facts themselves. So this is no longer just a challenge for the scientific community. It's a call to all of us to stand up for knowledge, truth, and reason. And with this in mind, I will start my presentation. So the goal of my lab is to study the role of neuroimmune crosstalk in host defense. we know that both the immune and the nervous system are able to sense injury and infection through the expression of multiple cell surface receptors. Upon activation, these two systems provide effector responses, both at the local and the systemic levels. And we are really interested in understanding how they cooperate to achieve these functions. Today, my presentation will concern two different models of skin damage. The first one is pathogen-free damage induced by UV light. It's a sunburn-like model. And the other one is a model of viral infection. In both models, we are studying how neuroimmune pathway promote tissue repair and protection. So as you know, the skin is a very important barrier tissue in which many immune cells are present or recruited, including macrophages, dendritic cells, mast cells, T cells, and also the recruitment of neutrophil and monocytes upon inflammation. The skin is also highly innervated by sensory neurons, and among them, nociceptive sensory neurons, which are stained here using the marker NAV1.8, can be very close to immune cells in the skin. These nociceptive sensory neurons, upon injury or infection, transmit signals to induce pain perception in the brain. But in addition, they also produce locally some mediators and neuropeptides that can directly act on immune cells. Several mediators have been identified to be involved in this neuroimmune crosstalk. However, depending on the pathological context, this molecule can have either pro or anti-inflammatory properties, and also this pathway can be either deleterious or beneficial to the host. So we really need to understand better how these pathways are regulated at the molecular level. So as I said, the skin is highly innervated by such sensory neurons. If we focus on the NAV1.8 positive sensory neurons, which are mainly nociceptive sensory neurons, we can distinguish two different families. The first one are peptidergic nociceptors, which are known to produce the neuropeptide CGRP and substance P. And another subset of so-called non-peptidergic nociceptors express other markers, including the marker GNIP, and produce other molecules like TAFA4 and glutamate. The cellular bodies of these neurons are located in the dorsal root ganglia, close to the spinal cord, as you can see here. And we can analyze their response, for example, here, by staining them with marker and monitoring the expression, for example, of the transcription factor ATF3. So in the first part of my presentation, I will describe the data we obtained in a model of sunburn. This study has been performed in the lab by Guillaume Eufel, a senior researcher, together with two PhD students, Guillaume de Broas and Pierre-Vincent Barbon. They set up a mouse model to study UV-induced skin damage and tissue repair. They found that skin overexposure to UV light induces the selective activation of a subset of sensory neurons, which express the marker GNIP, as you can see here. This neuron upregulates the transcription factor ATF3 after UV radiation. So to selectively study the role of this neuron in the tissue repair process, we use a genetic model in which we can selectively ablate these neurons. And here, as you can see, when these neurons are lacking, we observe an increase in the inflammatory response in the skin of these mice. And in the end, there is a tissue repair defect with a lot of fibrosis in the skin of these mice. So meaning that these GNIP-positive sensory neurons control inflammation and promote skin repair in this model. So we also analyze the molecular basis of this regulation. I don't know I'm not going to go into all the details of this study because it has been already published some years ago, but I will just summarize the main findings. We found that a subset of Guinea-positive neurons are activated after UV radiation and produce in the skin the neurokine TAFA4. TAFA4 can act directly on macrophages, and in particular in this context, they induce the survival of a pro-repair subset of TIM4 positive macrophages, and this molecule induces their production of IL-10. Together, this pathway is very important to promote skin healing and to induce the resolution of inflammation. And consistent with this, when we look in TAFA4 knockout mice, as you can see here, 35 days after UV radiation, these mice exhibit impaired skin repair and a lot of fibrosis. Interestingly also, if we compare this phenotype with the phenotype of mice in which we deplete the whole population of Guinea-positive neurons, we also observe this skin repair defect, but this leads to more severe skin lesion and greater impairment of repair, meaning that in addition to TAFA4, other neuroimmune pathways play a role in skin repair in this model of tissue damage. So to better understand what could be the other mechanisms, we hypothesize that in addition to the local regulation that I already described, there could be a role of the activation of the central nervous system in this model. We know that sunburn are painful and that the pain perception is activated, and we were wondering if other pathways could be activated. So to address this question, we use another model in which we can directly monitor the expression of cephos after UV light exposure in the brain and in different tissue. So here you can see the data. And as you can see, this skin damage triggers transcriptomic changes in several brain regions, including the paraventricular nucleus of the hypothalamus, which is shown here. You can see here in better way, and also here. The PVH is a major player in the hypothalamic pituitary adrenal axis, and it's known to coordinate body's hormonal response to stress. So this region is activated. Is it correlated with the production of endogenous glucocorticoid in the blood circulation? So this was our next question. And to test this, we exposed the mice to UV light, and we monitored the glucocorticoid response in the bloodstream. As you can see, after UV radiation, we could observe an increase in the glucocorticoid levels at day 1 and 2 post-irradiation. And very importantly, ablation of the Guinea-positive sensory neurons abolishes this glucocorticoid response. Importantly also, pain relief using buprenorphine in mice is also sufficient to limit glucocorticoid production. So UV-induced skin damage activates the central nervous system and stimulates glucocorticoid release into the bloodstream. So next, we wanted to know whether this pathway has an impact on the tissue repair process. So to test this, we use an inhibitor of glucocorticoid synthesis, metirapone, and we treated the mice with metirapone after UV radiation. And as you can see here, so this is the control showing that glucocorticoid levels are reduced. And inhibition of this pathway resulted in increased skin swelling, delayed skin healing, and delayed skin regrowth, as you can see here. These data were confirmed by using another inhibitor, ROU486, which is an antagonist of the glucocorticoid receptor, NR3C1. And as you can see here, the topical administration of this inhibitor also lead to tissue repair impairment. So to conclude for this part, we found that the sensory nervous system plays a pivotal role in skin repair by promoting the local release of the neurokine TAFA4 in the skin, and also by inducing pain perception, which is associated with systemic glucocorticoid production. Now I am going to switch to another part of my presentation, which is the study of the role of these neuroimmune pathways in resistance to infectious disease. We know that host survival to infection involve immune-driven resistance mechanisms involved in pathogen elimination, but it involves also tolerance mechanisms which preserve skin integrity. And it is really the equilibrium between these two, this kind of mechanisms that determines the overall outcome of infection. And this concept of disease tolerance is really interesting. It can be defined as the host capacity to maintain tissue function and to limit disease severity without necessarily reducing pathogen burden. And as you will see in the next slide, we found that the neuroimmune pathway are important to promote this mechanism of disease tolerance. So Anaïs Roger, PhD student, now postdoc in Canada, has studied the role of neuroimmune interaction in the context of herpes simplex virus type 1 infection. This virus induces a skin lesion, as you can see here, that can be quite painful. And similarly, in the mouse model, we can observe some very clean, similar skin lesion. But interestingly, this virus is also able to infect neurons and to replicate in the dorsal root ganglia, as you can see here. So to understand the role of nociceptive sensory neurons during HSV1 infection, we use a mouse model lacking the whole subset of NAV1.8 sensory neurons. So these mice were infected, and as you can see here, upon HSV1 infection, they develop very more severe and larger inflammatory lesion, as you can see here, and as it is shown on this graph. Importantly, this is not associated with a difference in viral load. The virus is similarly eliminated in both mice. By contrast, what we will observe is an increase in the recruitment of neutrophils in the skin of these mice, as you can see here. So we next wanted to understand in more details what could be the mechanisms of this regulation. To do so, we looked again in the dorsal root ganglia, and we tried to identify which neuronal subsets show transcriptomic changes upon HSV1 infection. And as you can see here, we found that both the peptidergic TRPV1 positive neurons and the GINI positive subset were activated in this pathological condition. So we decided to test then whether neuropeptides or neuromediator can be produced in response to HSV1. And especially we focused on these mediators which are known to modulate immune responses. So to address this question, we perform in vitro experiments by using a primary neuronal culture from DRG neurons, and we expose them to HSV1. By doing this, we observe a specific production in the supernatant of two mediators. First one is substance P, and the second one is TAFA4. We didn't see any specific, we didn't detect glutamate or CGRP in this model, showing that these two molecules can be produced by neuron upon HSV1 exposure. So first, we tested the role of TAFA4 in this model, and we looked whether this molecule was produced in vivo upon in vivo infection. As you can see, we were not able to detect TAFA4 in the skin of infected mice, and this is consistent with the fact that we didn't observe any clear phenotype tissue lesion difference in TAFA4 and control mice. By contrast, we identify a specific production of TAFA4 in the dorsal root ganglia. So we decided to focus on the analysis of the immune response in the DRG in these mice. So first here, it's just to show you in the course of HSV1 infection in a wild-type mouse, as you can see, there is a huge recruitment of immune cells in the DRG with an increase in the size of the DRG upon infection. And this is mainly mediated through the recruitment of inflammatory monocytes, which is consistent with the higher production of the chemokine CCL2. So we decided to test whether TAFA4 could be involved in the regulation of the immune response in the DRG. And so, as you can see here, in uninfected DRG, when we compare the number of CD45 positive immune cell in the DRG, they are similar in the two mice model, in the two mouse models. By contrast, so as you can see, upon infection, we observe a large increase in the recruitment of immune cells in the DRG of wild-type mice, and this increase was even higher in TAFA4 knockout mice. Meaning that we found that both macrophages and neutrophils are increased in the DRG of infected mice, and interestingly, this is observed after the virus has been eliminated. This increase in immune cell recruitment is associated with a decrease in IL-10 production. So it means that TAFA4 promotes IL-10 production and downregulates the inflammatory response in HSV1 infected dorsal root ganglia. So this is a summary of these results. We found that upon HSV1 infection, an immune response is initiated in the dorsal root ganglia, and after virus elimination, there is a very important role of TAFA4 in regulating IL-10 production and in promoting the resolution of inflammation in the DRG. And we are suspecting that this regulation could be important for reducing pain perception, and this is something that we are currently testing. As I told you also, we found that NaV1.8 sensory neurons are very important to control the neutrophil response in the skin and to resolve inflammation. This is required to promote tissue repair. We are also analyzing the molecular mechanisms behind this regulation. I have no time to show you the data, but as I told you, this is TAFA4 independent. So it means that the two independent neuroimmune pathways are involved in this model in the promotion of disease tolerance. And this defense strategy does not exert a direct impact on virus load, but it is essential to limit the negative impact of infection on health and fitness. Finally, I would like to highlight that the therapeutic potential of the TAFA4 molecule. This molecule has been shown to directly act on macrophages, and to promote macrophage, tissue repair, and anti-inflammatory functions. So given the very important role of macrophages in multiple human diseases, we think that this molecule could be a good molecule to use as an anti-inflammatory agent, and this is something we are trying to test also in the lab. So now I would like to thank all the people who participated in these studies. I already mentioned some of them, but many other team members were involved in these studies. I would like also to thank our collaborators, Aziz Mokrish, a neurobiologist involved in our studies, Franck Arboni, Laura Maquet, for the HSV1 collaboration, many other scientists at the Centre d'Immunology who help us in our studies. And I would like to thank also the funding agencies and our partners who funded our studies. And I would like to thank you for your attention.
Thank you very much for this excellent presentation, Sophie. Can I maybe start with one question which came up to my mind when you talked about disease tolerance? How much actually is age influencing disease tolerance, having in mind that this is a process of pro-inflammatory responses ongoing? And if we think on other herpes viruses like herpes zoster that is increased in age, there is actually, you know, maybe other parameters that promote the disease. It wouldn't fit really with the IL-10 hypothesis you are posting. Is it a matter of different herpes viruses, how they are, how the defense mechanisms are working, and how in general is age counteracting or promoting this fact of disease tolerance?
It's a very good question. We didn't check this parameter in mice. It could be what we monitor in our study is really the first stage of infection, the primary infection in this model, in this mouse model. Your question is more related to the reactivation of the virus, which is maintained in the nervous system and can be in humans and can be reactivated upon stress or other parameters. So it's difficult to answer your question. I think this pathway is very important, at least for the primary infection. Okay. Then for the reactivation of the virus, it's more difficult because the mouse is not a very good model.
Good model for that. Okay. And on the other hand, when you think of other parameters in defense mechanism like CD8 cells or something like this, have you been looking here on the importance of those cells?
Yes, exactly. I didn't have time to show you, but we also monitor the CD8 T cell response in these mice, and we found that when the sensory neurons are absent, there is a huge neutrophil influx in the skin, and this is associated with an impaired CD8 priming in the skin draining lymph node. So the adaptive immune response is also affected, and the CD8 T cell response is also affected. And we are currently analyzing the molecular basis of this inhibition.
Thank you. We have several.
We have several questions from the public, from the people who are listening to your fantastic talk. And there is one that is, I think, a bit difficult, but do we have a system, a readout in the blood to understand whether our immune system in the brain is okay, it works? I know it's a very difficult question. You know, can we have a biomarker to identify whether everything is okay, whether we are developing a disease?
Yeah, so one of the markers that we used in this study is the monitoring of glucocorticoids. Glucocorticoids can be a readout of activation of the hypothalamic pituitary adrenal axis. And when this pathway is activated, it induces the production of glucocorticoid by the adrenal gland. And glucocorticoids are very important in regulating immune responses and many other biological processes. So it could be one readout. Otherwise, it's very complicated because during infectious diseases, as I said, there is a balance between resistance mechanism and tolerance mechanisms. And inflammation is very important for pathogen elimination, but must be also controlled. So it's easy to have a clear marker to monitor the ways disease can evolve, because it's really a balance and it's highly dynamic.
And then we have several questions actually about tumors in the brain. You know, does our immune system does something against when a tumor is raising? You know, what can we do? What does our immune system?
This is not my expertise. No. I think it's another question that we are not addressing in the lab, but I think it's a very important question. Yeah.
Yeah. Thank you.
To be able to promote immune response against tumor in the brain, and I think it's not easy.
Thank you very much, Sophie. It was an excellent talk and very inspiring. Thank you so much. And I just
Thank you very much.
Thank you for being with us, and maybe you stay on a little bit more. Just all the people who are listening, please use the chat to ask your questions, because we can take them online. Only for the next talk it's not possible, because we have only a recorded talk by Professor Stefan Hauser, who is unfortunately at the same time at another conference and has given us a recorded talk. But I would just like to introduce him shortly to you. Stefan Hauser is a neurologist and neuroimmunologist, and he's professor of neurology at the University of California in San Francisco. He has been working years on the identification of genomic and epigenomic background of multiple sclerosis, and started with a big project in 2011 to identify multiple sclerosis associated risk alleles. By now there is nearly an entire array available for common variants associated with multiple sclerosis susceptibility. But he is also has been working on B cell and the immune responses and the pathogenesis of B cells for multiple sclerosis, and was one of the first person who's used rituximab, the monoclonal antibody against anti-CD20, as a treatment strategy against multiple sclerosis. And this is what he will talk about. The topic is on the immunological path to a cure of multiple sclerosis, and we will now listen to his recorded talk.
Hello. I'm delighted to join you at this international symposium commemorating the Day of Immunology. My name is Steve Hauser, and I'm located in San Francisco on the west coast of the United States. I'm going to speak about multiple sclerosis, a chronic multifocal central nervous system disorder caused by autoimmune-mediated myelin loss. There is progressive neurodegeneration, strong genetic predisposition with more than 230 variants, including HLA as causative to risk, and one variant now identified as causing a severe expression of the disease. There is a strong influence of environment, with Epstein-Barr virus a likely culprit. The prevalence of MS has increased, and currently in the United States, approximately 1 million people are affected. The prevalence has increased especially in women over the past century. And as you'll see, remarkably effective treatments are now available. Similar to other chronic autoimmune conditions, MS is thought to have 3 principal phases: a premonitory preclinical phase, an inflammatory tissue destructive phase, and then a late smoldering degenerative phase. MS typically begins with relapses followed by remission. This happens in about 90% of patients. Attacks occur approximately once every 9 months and manifest as interruptions of long pathway signaling in the brain: double vision, visual loss, weakness, sensory change, incoordination, bowel or bladder difficulty, among others. The MRI era has taught us that bursts of focal brain inflammation occur 7 or 8 times more frequently than do clinical attacks. Most areas of inflammation occur in silent regions of the brain. So approximately every 6 weeks, a typical patient with recent onset MS will have a burst of brain inflammation. We initially thought and hoped that if we could silence relapses with a high efficacy therapy, we would eliminate the neurodegeneration and slow progressive disability during the relapsing phase of MS. And we're not sure if the relapsing phase would ever evolve into gradual progressive MS with non-ambulatory status in most patients. But what we learned, unfortunately, is that when we began high efficacy therapy, we unmasked a slow neurodegenerative process that was actually present from the earliest phases of disease, but was previously obscured by relapses and remissions. Thus, relapsing MS and progressive MS are both occurring throughout the life cycle of the clinical disease. I'll now discuss briefly each of these phases. First, the premonitory preclinical phase. Serologic autoantibodies have been the most effective biomarkers by far to define the presymptomatic phase of autoimmune diseases. For example, in systemic lupus, initially there is the appearance of relatively benign autoantibodies, ANA, anti-Rho, anti-La, followed subsequently over years by the appearance of more pathogenic autoantibodies, anti-double-strand, SM, and RNP antibodies. Similarly, in type 1 diabetes, the initial appearance of a single autoantibody, either GAD65 or insulin antibodies, is followed by the appearance of the second antibody and then additional ones over time. In rheumatoid arthritis, citrulline-aided antibodies followed by other bad actors over time also occurs. The timeline is quite different. In lupus, it's about 9 years from the first autoantibody to clinical disease. In rheumatoid arthritis can be several decades, but in type 1 diabetes is quite short. In pediatric onset type 1 diabetes, the timeline for the spread of these autoantibodies occurs in less than a year, approximately 9 months on average. This short timeline has set the stage for efforts to treat the disease before it begins. And many of you may know that teplizumab, the anti-CD3 monoclonal antibody, has been shown in initial studies to prevent the appearance of glucose intolerance in type 1 diabetic prone individuals who harbor more than one diabetes-associated autoantibody. Until recently, however, there were no specific autoantibodies identified in people with MS. That has changed over the past year with the publication of work by a UCSF group led by Dr. Michael Wilson that identified a humoral immune signature specific to a subset of MS patients that appear years before diagnosis. This was enabled because of the Department of Defense Serum Repository, which is a huge biorepository of more than 81 million serum samples stored from people who entered the service and their family members and maintained longitudinally. And in the data set that was used, 250 individuals who developed MS but did not have MS at the time of the first blood draw at a mean of 5 years before clinical onset could be interrogated. The method uses a phage display library to probe antigen-antibody interactions by immunoprecipitation from serum or CSF. It's a whole human proteome approach that tiles 49-mer peptides across all 23,000 proteins in the human proteome. And it is a very powerful method to search for antigen-specific, disease-specific sequences. The first lesson that emerged is that the antibody signatures within the individuals were remarkably consistent over time. But even more exciting, a distinct immunogenic cluster emerged that included only MS patients before and after onset. Enrichment was from a clear but disparate group of enriched peptides belonging to more than 50 different proteins in the pre- and post-onset samples. And alignment of these enriched peptides from the subset of patients revealed a distinct protein motif characterized by an initial proline and then a serine-arginine repeat. The motif was similar to that contained in a variety of infectious agents, and in fact had the highest homology to the BRRF2 protein and glycoprotein M protein of Epstein-Barr virus. These changes were associated with increase in serum neurofilament levels suggestive of ongoing brain disease even at the pre-symptomatic stage, was present in 12% of patients, but in 30% of patients with familial MS. It is identical to an EBV-specific signature identified in earlier studies of MS antibodies. And I'd also mention that for individuals with this signature, the antibody titers are concentrated in the spinal fluid compared with serum. So for the first time, we now have a specific, if insensitive, antibody signature for study of MS in the pre-symptomatic phase. I'll turn now to Act 2, the inflammatory phase of MS. The anti-CD20 monoclonal antibodies have revolutionized the treatment landscape for MS. They have proven to be not only highly effective, but also extremely well tolerated, especially the humanized and human monoclonal antibodies, ocrelizumab and ofatumumab, which are both in widespread use today. And approximately 1 million patients are receiving these monoclonal antibodies today worldwide for people with MS. For ocrelizumab, where we have 12-year safety data, it is very clear that the safety profile remains outstanding, and that is probably because with these treatments that target just the CD20 antigen present at the mid-differentiation stages of B lymphocytes. There is preservation of stem cells and pro-B cells, ensuring B cell reconstitution after treatment. And because plasmablasts and plasma cells are also CD20 negative, long-term immune memory is also preserved. The benefits of treatment with anti-CD20 B cell targeting monoclonal antibodies in MS are truly extraordinary. These are the results during the pivotal phase and the open label extension with ocrelizumab, the humanized monoclonal antibody in widespread use worldwide for people with MS. In the pivotal trial, ocrelizumab was tested against beta interferon, a platform therapy at the time in widespread use for people with MS. The readout here are areas of MRI inflammation, a surrogate for clinical relapses. Here with interferon, you can see that during the 2 years of double-blind treatment, there was a reduction in brain inflammation measured by MRI of approximately 75%. With ocrelizumab, however, the effect size was greater than 99%. This has been called the greatest effect size apart from antibiotic trials in modern medicine. They are truly remarkable findings of a complete cessation of brain inflammation. During the open label extension studies, patients on interferon are then transferred to ocrelizumab, or ocrelizumab patients remain on the medication. And you can see that the cessation of brain activity, of inflammatory brain activity continues. In terms of clinical relapses, after a decade, patients on average have less than 1 relapse per lifetime. So relapsing MS is truly now in the rearview mirror. I'll now turn to the third neurodegenerative phase. Now let's turn to accumulation of disability in the ocrelizumab trials. And as you can see here, in this population of patients with relapsing MS, the proportion of patients who accumulate disability annually is about 4% per year on the interferon platform therapy, and that this is reduced by a little bit more than 50% in patients on ocrelizumab. Furthermore, when all patients receive ocrelizumab during the open label extension, those patients initially assigned to interferon don't catch up. And this is a truism throughout the clinical trial experience in MS. Early disability is not recoverable with later onset of high efficacy therapy. And this has changed the treatment paradigm worldwide in favor of treating patients with high efficacy therapy at the beginning of the disease whenever possible. The biology responsible for these extraordinary effects of B-cell therapy on MS is also now coming into focus. Initially, it was thought that autoantibodies were likely the explanation for B-cell mediation in MS, but the very rapid clinical response argues against autoantibodies being the primary culprit. In people with MS, B cells are polarized towards a pro-inflammatory phenotype with excess secretion of TNF-alpha, IL-6, and other pro-inflammatory cytokines. This is likely part of the story, with more pro-inflammatory cytokines generating bystander inflammation as well as neurodegeneration. Remember that 95% of us have been infected by Epstein-Barr virus during our lifetime, and that B cells, and memory B cells in particular, are the lifetime reservoir for Epstein-Barr virus in most of us. So perhaps the fact that B cells are harboring Epstein-Barr virus and are triggered by lytic cycles of Epstein-Barr virus towards B cell activation could be part of the story, but likely not the whole story either. It is now very clear that B cells that are resistant to peripherally administered large monoclonal antibodies that don't cross the blood-brain barrier effectively are also present in the brains of people with MS, particularly in the deep layers of the meninges. And these B cells may be the reservoir for the toxic properties of cytokines and other molecules such as free radicals secreted by B cells in people with MS. B cells are also having important crosstalk with microglial cells, which are clearly part of the pathology of chronic MS. But most of the attention has focused on the antigen-presenting function of B cells as perhaps explaining the therapeutic effects. As you know, B cells are professional antigen-presenting cells. They can pick up antigen on their surface B cell receptor that is then endosomally processed and presented to T cells that can then activate these responding T cells. And it is now thought that in MS patients who are likely to have specific HLA genes, especially the DRB1-1501, what we used to call the DR2 HLA haplotype, that these class II molecules are presenting fragments of the class II molecules themselves, activating T cells through low avidity, that then through higher avidity, the identical T cell receptors can be reactivated by a variety of other antigens, including antigens of Epstein-Barr virus, Akkermansia bacilli, also epidemiologically associated with MS, as well as myelin proteins. So the HLA molecules themselves may be molecular mimics for spread of an autoimmune response. Thus, we now understand that there are multiple different pathologies of MS, one being the classic focal inflammatory plaques, comprised of both B cells and T cells, with CD8 T cells usually at the leading edge. These meningeal B cell rich but also T cell containing follicles, these microglial lesions at the edges of old plaques called chronic active plaques that are slowly expanding and destroying tissue from within, and then a diffuse microgliosis, CD8 cytotoxic T cell infiltration, and astrogliosis that accompanies late disease. And with our treatments, we are completely eliminating the B cells that are responsible for these focal lesions and that are present in some of these meningeal aggregates. But these other pathologies appear to be driving late progressive MS and remain the major therapeutic targets for future clinical data. They are certainly the areas of greatest unmet need today, patients with advanced progressive MS. So what are the current prospects for more complete disease suppression, especially for patients with progressive MS symptoms? The first, of course, are next-gen B-cell therapies. And especially here, I would highlight Bruton's tyrosine kinase inhibitors. Highly selective BTK inhibitors are in late-stage clinical trials and will unblind, I expect, in early 2026. And CAR T therapies, as with other autoimmune diseases, very preliminary data suggest the possibility of extraordinary benefits in people with MS. Targeting Epstein-Barr virus is another very active and exciting area. As I mentioned earlier, early treatment will almost certainly be the most effective. And in late disease, multiple cell types are likely additional culprits. And here microglia and CD8 T cells are targets for future therapeutic trials. The progress that I've been fortunate enough to witness against MS has been truly extraordinary, but much work remains to be done, both against MS and other autoimmune diseases. And as the older generation passes the baton to the younger, and I hope there are many people in that latter group in our audience, we are beginning a paradigm shift away from a hope to just suppress disease to attempts to cure or even prevent chronic autoimmune diseases. We are moving from graduated therapeutic strategies to the use of highly effective strategies as early as possible, understanding optimal duration of therapy, and then rather than lifetime therapy, considering off-treatment monitoring protocols with biomarkers that are highly effective to predict the reappearance of disease. I think the future is going to be the most exciting area in medicine. Huge thanks for inviting me to be part of this wonderful afternoon.
Thank you very much to Stefan Hauser. It was an excellent, wonderful presentation, and we wished we could discuss with him, but this is today not possible, so we just send our greetings and our thanks for this excellent presentation. And I think before we go in a short break, we just want to say that there are a lot of hellos from all over the world, and we want to greet people from Malawi, from Nairobi, Kenya, hello from Belgium, from Brazil, from Greece. So I hope even more from all over the world are just with us and also stay on with us. We will have a break now and meet each other again sharp 4:20. See you soon, and just look to the nice videos that are coming up now. Thank you.
This year, April 29th, marks the Day of Immunology. This year's theme is Brain and Immunity, Immunological Insights into Neurological Disorders. And once again, EFIS, UIS and Young EFIS have joined forces to organize a variety of activities to celebrate this important occasion.
We're excited to kick off the Thank You Immunology social media campaign today. Throughout the week, we'll be sharing short videos from EFIS and Young EFIS representatives leading up to the 2025 Day of Immunology. Our aim in this campaign is to spark public conversation around the critical role of immunology in our society and our daily lives. We invited our network to share videos highlighting initiatives planned by EFIS and Young EFIS societies for the 2025 celebration, as well as personal reflections on what immunology and the Day of Immunology mean to them.
And as part of the celebration, we also warmly invite you to join us on April 29th for the webinar, which is promoted together by UIS and EFIS. In the webinar, we will explore how the immune and nervous systems interact and discuss immunological mechanism and treatment strategies for the increasing burden of neurological diseases such as multiple sclerosis and Alzheimer's disease. And you will find all the details and registration info on the UIS and EFIS website.
I take the opportunity of this invitation for the Day of Immunology for another invitation. On behalf of IOS and the Austrian Society for Allergology and Immunology, I'm delighted to invite you to Vienna for the 19th International Congress of Immunology, which will take place in August between the 17th and the 22nd of this year, 2025. Under the theme Waltzing into the Future of Immunology for a Better Health, the Congress will showcase cutting-edge research and global collaboration in our field. Join us for exciting scientific session, but also to enjoy the unforgettable experience of discovering Vienna rich history and also vibrant present. All the details are available on our internet page, iois2025.org.
We're looking forward to celebrating the 2025 Day of Immunology with all of you on April 29th. And don't worry, if you miss anything on social media, all videos will be available on the Day of Immunology YouTube channel.
Enjoy the Day of Immunology and join us for the campaign #ThankYouImmunology.
Thank you, Immunology.
Thank you, Immunology.
Hello, everyone. I'm delighted to send this message on behalf of the Japanese Society of Immunology in the celebration of the Day of Immunology. We celebrate the Day of Immunology every year by holding public event Meneki Fushigi Mirai in Tokyo, introducing the importance and attraction of immunology for everyone from children to adults. The main attraction, an event called Meneki Fushigi Mirai, meaning Amazing Immunology for Future. Over 2,000 people join this event every year in the Tokyo Science Museum. This is actually the highest number of visitors of all scientific events held at the museum. During their visit, people can listen to talk about the immune system, chat with immunologists, and even do experiments such as making tissue from slime, observing blood cells under the microscope, and preparing DNA from banana with detergent and disinfectant. Immunology is important to us and our community because we believe that it's one of the most important scientific fields for the public from the view of overcoming infection, cancer, allergy, and autoimmunity. In December, we will hold an annual meeting in Himeji, close to Kobe. Please join in the nice meeting at a wonderful place. Thank you.
Immunology lovers, get ready! The Day of Immunology 2025 is here, and this year we're going big, celebrating the incredible 100th years anniversary of Jaroslav Sterzel, the founder of Czech Gnotobiology.
Join us on April 29th in Prague for a day packed with cutting-edge science, fascinating discussions, and a deep dive into the world of germ-free models. Kicking off with Gnotocon, we honor Sterzel's legacy with insightful talks from leading experts.
Wait, what's this Gnotocon thing?
That's the exciting Gnotobiology conference. In the keynote lectures, we will explore the history of Gnotobiology established by Professor Sterzel and even uncover how bacteria talks to our immune system. No brain required. Plus, hear short talks from researchers working with germ-free models, pushing the boundaries of immunology.
But wait, there's more. The CYI Mixer will blow your mind, literally. We'll dive into the theme, Brain and Immunity, with exciting lectures, an AI in Science panel discussion, and chill zone for winding down.
Great science, great company, and a toast to 100 years of groundbreaking immunology. What's not to love?
Join us in Prague or follow along online. Let's celebrate immunology together. Thank you, Immunology.
CIMSA, a regional federation under IUIS, unites 12 national societies across Asia-Oceania. Immunology drives scientific progress and is essential for public health and strong healthcare system in a diverse region. CIMSA supports this by hosting congresses and training courses that promote collaboration and education. Through these events, we aim to empower communities and build a healthier future for all.
Hello everyone, my name is Bernardo Silva. I'm a PhD student at SEM and I'm the newly elected spokesperson for the Next Generation Immunologists, and today I'd like to celebrate the Day of Immunology, which I think is a very important initiative since as immunologists we also deserve some recognition from time to time, and I will be doing that by reading up on my favorite subset of cells, T cells.
Hello, my name is Kveta and I'm the newly elected vice spokesperson of the Next Generation Immunologists. I study the immune and stromal crosstalk in the context of granulomatous disease. And I'm excited about the Day of Immunology this year because it gives our new Next Generation Immunologists board an opportunity to introduce ourselves to the immunology community.
Hi, my name is Taras. I'm studying immunology of liver disease. I think immunology is important because it teaches us how to support our immune system and how to be a better partner for it when it's facing a hard time fighting cancer or pathogens or during autoimmune diseases or allergies.
Hi, I'm Laura. I'm a PhD student at the Medical University of Vienna and at SEM, and I'm studying the immune system of the skin. And I think the Day of Immunology is a very important initiative to raise awareness for our research.
This is where it's at.
The brain is a very complex organ. It's one of the last frontiers in drug discovery research. This is where the science is breaking. The immune system is a major part of the pathology of many brain diseases, and intervening at the level of immune signaling can be really impactful in disrupting disease progression. As a matter of fact, there is hardly a brain disorder in which the immune system is not altered in some way. So, that opens up huge potential. On top of that, human genetics has told us that the immune system can be responsible of the manifestations of many brain diseases. In many neurodegenerative diseases, for example, multiple sclerosis, Alzheimer's, Parkinson's, and rare form of Parkinson's, the science has progressed and therapies have to follow. So the ability of us to treat neuroimmune diseases is bigger now, more than ever.
I de værste perioder, der har jeg da haft 23 anfald på en måned.
I can get dizzy, nauseous sometimes vomiting.
It feels like light is like burning.
Migraine has a huge impact on my life. It affects my life in every possible aspect.
It hurts to even talk, dizzy,
burning,
nauseous, non-existent.
You can't understand what it's like to be in constant pain.
Hi, we are here from the Dutch Society for Immunology, and I'm interviewing people to ask them, what does immunology mean to you?
Immunology is my life. It's the only thing I think of when I get up and when I go to bed. It's everything.
Immunology is challenge, it's fun, and it's work.
Immunology is complex, I would say.
And that's why we make it even more complex by connecting it with metabolism. And I guess then it's the basics of life, being healthy or being sick, and that's what we like a lot, especially.
Yeah.
And that's
Immunology means hope, I guess, or a powerful weapon, if you will. That's how it means the ability to take what's already there and utilize it and make it into a powerful weapon. That's it.
So for me, during my PhD, I figured out it's very much a double-edged sword, and it's very interesting to see how can we find the positive things of immunology to fight pathologies of different diseases.
Immunology is what keeps us alive. Without immunology, we would all have passed away years ago.
Puzzles.
A fun job. How?
Immunology to me means actually a consortium of many different cells that cover all of our body. So it's not like an organ which you have resident cells, but in immunology you have different cells that patrol our body, and they have to communicate with each other. They have to work together. And it's like a big consortium, as we do in research.
Dr. Kate Connor, and I will be celebrating the 2025 Day of Immunology here in the discipline of Physiology in Trinity College, Dublin. I will be using electrophysiology to study the role that immune cells play in driving brain tumour-related epilepsy. Immunology is incredibly important to the brain tumour community, and studying the role of immune cells in driving brain tumours allows us to understand the mechanisms which underpin brain tumour progression, and also allow us to develop targeted precision medicine strategies for patients with this condition.
Hi, my name is Dr. Emer Mylad, and today I celebrate 2025's Day of Immunology here in the research labs at the School of Biochemistry and Immunology at Trinity College Dublin in Ireland. I'm exploring the impact the cancer environment has on natural killer cells so that we can design effective immunotherapies.
Hello, my name is Dr. Melissa Conroy, and this year I'm celebrating the 2025 Day of Immunology by teaching neuroanatomy here in the discipline of anatomy in Trinity College, Dublin. And as we teach neuroanatomy to our students, we also like to highlight neurological disorders such as metachromatic leukodystrophy, which affects the brain and also the spinal cord.
My name is Dr. Laura Kane, and I think that immunology is important because it helps us address mechanisms underpinning several diseases, including cancer. Working in the Department of Surgery in Trinity St. James's Cancer Institute, we're researching how the immune system is dysfunctional in pancreatic cancer and aiming to develop immunotherapies to restore this immune function in cancer and boost tumour eradication.
Immunology is important because every day we encounter countless microbes. Most of them are harmless, but others can cause diseases, and it's our immune system that works to protect us.
Our immune system is important to combat infection and keep us healthy. However, sometimes it can malfunction and cause autoimmunity. That's why it's important to study immunology to help us understand these problems and find best ways to tackle them.
But immunology isn't just about an individual's health and medicine. It affects the whole community and even the world with impact on public health. So, the more we understand how the immune system works, the better we can fight disease and improve human and global health.
To celebrate the Day of Immunology, we are hosting a scientific meeting organized by the newly formed Young Portuguese Society for Immunology group. This will feature presentations from young researchers in development, infection, neuroimmunology, autoimmunity, and tumor immunology. This meeting will act as a platform for scientific exchange, bringing together the Portuguese immunology community to foster collaboration between everyone.
Hi everyone, so I'm Angeliki and I'm a current board member of the Young Immunologist of the German Society of Immunology, and I would like to share with you guys why for me immunology is super interesting and important. You have to imagine that in many societies childs and humans still die from immunological disorders or pathogens that are so much in control here in Europe and the Western society. Imagine what is happening in your body when you have an autoimmune disease and your immune system is out of control. Understanding all of this and what impact the cells in your own body have by saving your lives but also contributing to making you sick is something that is extremely fascinating to me and is so multiplex that we need to understand it in order to contribute to a better health system and care system for us, but also for poorer countries. So please share whatever you know and love about immunology.
Hi, I'm Bianca. I am a PhD student in Heidelberg and also a member of the Young Immunologists at the German Society for Immunology, the DGFI. And for this year and the Day of Immunology, our monthly seminar series will feature an expert in the field of neuroimmunology, Michael Kilian, who also recently won a prize here in Germany for his research in GBM. So if you want to join our initiative, just follow us on our social media channels to find all the latest info. And together with the DGFI, we are also actively creating numerous posters on different topics of immunology that you can find all on our website. And also there are a couple of games for the smallest of us if you want to get your kids involved in the exciting field of immunology.
Have a good day of immunology, learn as much as possible. Bye!
Since the immune system is our main protection against illnesses in general, immunology is an important field of study for the general population that help us understand how our bodies react to bacteria, viruses and other diseases which directly affect public health. Immunology plays a crucial role in medicine, especially in developing vaccines that have helped control diseases like smallpox, polio, and more recently COVID-19. It also helps us understand autoimmune disorders where the immune system mistakenly attacks the body, as well as allergies caused by overreactive immune responses. This knowledge has led to life-improving treatments. Additionally, immunology is transforming cancer treatment through immunotherapy and is essential in organ transplants by reducing rejection risk and improving donor-recipients compatibility. Ultimately, immunology is not just about medicine, but also about collective well-being. Thanks to this science, we live in a safer world with increasingly effective tools to prevent, diagnose and treat numerous diseases.
For the International Day of Immunology on April 29, the Italian Society of Immunology has decided to highlight the importance of scientific research in the field of neuroimmunology that studies the relationship between immune and central nervous system aiming to understand the pathogenesis of different diseases, like multiple sclerosis. Researchers will be invited to select a specific scientific contribution in the field of neuroimmunology, and the most outstanding ones will be analyzed by competent SICA members that give us an overview about them. The goal is to raise awareness within the scientific community about this crucial branch of immunology.
Welcome back to the Day of Immunology event from Vienna. And we have our next speaker now, who is Maria Rescigno. Maria Rescigno is full professor of pathology in Milano at the University Humanitas. Maria has a lot of different things to do because she is also the deputy scientific director of the Humanitas Research Hospital in Milano, and she has a group. She's working on mucosal immunology and microbiota. Her first important discovery was that dendritic cells take up bacteria from the gut and trigger then the immune response to these bacteria. She also showed that the gut has a vascular barrier that resembles a brain barrier. And today Maria will talk. That was actually the first discoveries, but she did a lot of very important things working on the microbiota now and on the metabolites of microbiota. So today Maria is talking about the gut microbiome and neurological disorders. Her presentation is registered, but remember that Maria is online and she will be able to answer to all your questions. So, sia Maria Rescigno and her talk. Please.
So good morning, everybody. Today I will be talking about the gut microbiome and neurological disorders. So these are my disclaimers. Okay, so we know that the microbiota is involved in several functions of our body, including the food breakdown and immune system maturation. But recently it has been shown also that it's very important for brain development and neurological disorders. So first of all, I would like to just to summarize where the microbiota lives and what are the major barriers that we have to protect ourselves from the microbiota. So the microbiota is present primarily in the terminal ileum and in the colon, and it's here that we have two different layers of the mucus. So you have the epithelial cells, which are the first line of defense of cells of our body, that are here, the nuclear here in blue. And then the goblet cells and the epithelial cells are produced in the mucus layer, which comes in two flavors. So one that is very close to the epithelium and is a very tight mucus. And then you have a more detached mucus, which is also more permissive, and this is where the microbes can penetrate. So you see the microbes in green, and the mucus it's in red. This is one of the most abundant mucin, which is called MAC2 in the colon, and you can see that this mucin is in a network that is permissive for microbes to penetrate, while here that is closer to the epithelium, this mucus it's very strict, and so does not allow the microbes to penetrate and to attach to the epithelium, and so to avoid an ongoing inflammatory response. Then just below the epithelium, we have an additional barrier, which is we described some years ago now, it's the gut vascular barrier. So you have the mucus, you have the epithelium sealed one to the other by tight junctions, but then you also have endothelial cells that are organized in a barrier. And this resembles quite a lot the blood-brain barrier in that you have endothelial cells which are sealed one to the other by tight junctions. Then you have pericytes and enteric glial cells, which are the equivalent of the astrocytes. And altogether, these are making these endothelial cells very difficult to penetrate. So if microbes can cross the mucus, because they can degrade the mucus, they can enter and invade the epithelial cells, then they find an additional barrier which blocks them from from penetrating into the liver or in the brain via the portal circulation, and so then the blood circulation. And so there are some microbes like enteropathogens which are capable also to evade these defense, and so then to disseminate systemically, but this is not the case of many of the microbes present in the gut. So a steady state conditions, then just to summarize, we have these two mucus layers. So we have the outer mucus layer, which is the one that is colonized by microbes, and then the inner mucus layer, which is blocking these microbes from penetrating and for getting in close contact with epithelial cells. However, what happens during inflammation, this is the beautiful work of Sara Carloni, then this mucus is compromised, and so the microbes are not only found in this area, which normally would be empty of bacteria, but then you can find them also within the lamina propria because they can penetrate. And then what happens? So we know that during this condition, which is called leaky gut, which is a characteristic of some inflammatory bowel disease, but also other conditions, then 40% of these patients, they suffer from depression. And so the question that we asked is it possible that when you have an inflammatory response, which is leading to a change also vascular permeability in the gut, so an increase of permeability of the gut vascular barrier, that this ends up with a change in permeability of the brain, and so this may be responsible for the depression state of these patients, and not just for the fact that it's very difficult for these patients to cope with such a debilitating disorder. So what we looked at was at the gut vascular barrier permeability. So we use this marker, which is called PV1. When this marker is expressed, this means that endothelial cells are more permissive because they become fenestrated, and so they allow also the passage of large molecules. And so you can see that in healthy conditions, there is no marker. So because the barrier is organized in a barrier, but if you look at patients with ulcerative colitis, so IBD patients, then this marker becomes visible not only in the ulcerated area, but also in the distal area. So meaning that in the whole colon, you have an increased permeability of the gut, not only at the level of the epithelium, but also at the level of the endothelium. So then we looked at whether this is true also in a mouse model of colitis, which is the DSS model. So we gave DSS for 5 days to mice, and then we looked at different time points, the status of the gut vascular barrier. And so you can see here that already at day 1 after DSS treatment, so in the early phases of inflammation, then you already see the coming up of these markers, so meaning that these vessels become fenestrated. So this is a very fast event occurring during an inflammatory response, even before the change in epithelial permeability. So here you can see the ZO1, which is a marker of tight junctions. And so you can see that this is still present at day 1, and then at day 3 you see very little of it, and then at day 5 it comes back again. So just quantifying in different mice, you can see that the changing gut vascular permeability is a very early event, so occurring already day 1 when there is no change yet in the ZO1, so in the tight junctions. But then over time, the epithelium starts sealing again, so increasing the expression of ZO1, while the endothelium remains very permissive. So during an inflammatory response in the gut, there is an increased permeability, which is occurring, that is a very early event, both in the epithelium and in the endothelium. So then we wondered whether this was correlating with a change in inflammatory profile of these organs. So in the gut, we could see that at day 5, there was a recruitment of several inflammatory cells, including monocytes, neutrophils, and macrophages. And then we looked both in the liver and in the brain. We found that with the slightly different kinetics, also in the liver, we could see eventually an increase of monocytes and neutrophils, no changes in macrophages, while when we looked in the brain, we could see again an increase of monocytes and macrophages, but actually we could see an even reduction of neutrophils. So there are some fluctuations that are time-dependent and organ-dependent when there is an inflammatory response which is ongoing in the gut. So then we asked, going back to our original question, that was, is there a change in permeability of the brain? We decided to induce intestinal inflammation in the gut by providing the DSS for 3 days, and then we removed the stimulus and we gave water to the mice, and so to see whether this acute inflammatory response, whether it was then changed over time when you were removing the inflammatory stimulus. And so we provided to the mice a very large dye, which is a 70 kilodalton dye that we know is unable to penetrate the blood-brain barrier. And so our original hypothesis was that during inflammation in the gut, there was an increased permeability in the gut itself, and this was correlating with increased permeability in the brain. But we made two very unexpected observations. The first one was that this dye can already penetrate the brain before inflammatory response. So there are regions of the brain which are permissive for the entrance of this very large molecule. Then we could see a slight increase of permeability at day 1 after DSS colitis, but then we could see a complete shut-off of brain permeability at day 3. So suggesting that rather than having a higher permeability of the brain during an inflammatory response, we actually had a reduced permeability of the brain. So the brain is more permissive at day 0 than at day 3 after an inflammatory response. And this is a transient because at day 5 we could see again a restored permeability of the brain. So then we looked at which area of the brain was this dye was permeating. And we found that the area of the brain where the dye was permeating was the choroid plexus. So it was not the classical blood-brain barrier as expected, because this was not permissive, but it was the choroid plexus. And so what is the choroid plexus? So the choroid plexus is the structure present in 4 portal of entry of the brain, which are connected with the ventricles, and the choroid plexus is organized in a way that is so similar to the intestinal barrier. So we have an endothelium, and then we have an epithelium, but differently from the gut, where the external world is facing the epithelium first and then the endothelium, and both are very highly sealed in the gut in order to avoid the penetrance of unwanted molecules. The choroid plexus is different because it has to filter the blood and to generate the cerebrospinal fluid. And so the endothelium is very permissive, and the epithelium is the only one that is creating the barrier, but still it's permissive because it allows even large molecules to penetrate into the cerebrospinal fluid. And this allows to bring nutrients and mediators from the periphery into the brain to communicate with the brain. So this is an area of the brain which is highly permissive as compared to the blood-brain barrier, but still in structure it's similar to the intestinal barrier. And so you can see here in this 3D reconstruction that you have the epithelial cells sealed one to the other by tight junctions in the outside, and then you can see inside the vessels here, and these vessels are fenestrated because they express this PV1, which is the marker of fenestration. So these vessels are different from those in the gut because they are permissive, and so this allows the brain to interact with the rest of the body. So then we looked at whether the dye was indeed penetrating in this area. So the dye is now in violet. You can see at day 0, when there is no inflammatory response, that the dye can penetrate into the choroid plexus. At day 1, you see even more dye penetrating. And then at day 3, you can see that the dye is not there any longer. So meaning that indeed these vessels become highly restrictive in what penetrates. And this is transient because day 5 you can start see the dye penetrating again. The dye doesn't remain in the stroma of the choroid plexus, but it enters the cerebrospinal fluid. As you can see here, day 0, you can see the dye penetrating into the cerebrospinal fluid. Day 1, there is more penetration, and then at day 3, there is a high reduction of penetration, and then day 5, there is again penetrance of the dye. So the choroid plexus is the structure which is connecting the brain with the rest of the body and is highly permeable. But what happens during inflammatory response, that the PD-1, which is the marker of fenestration, which is normally present on the vessels, is completely downregulated at day 3. So during the acute inflammatory response, these vessels are not fenestrated any longer, and so this is why they don't allow the dye to penetrate. So to sum up in this part, in the choroid plexus, this is a region which is interacting, allowing interaction between the brain and the rest of the body. So they receive signals coming from the periphery, and these signals, if they are not dangerous, then they are transferred according to their size across the epithelium, and they reach the cerebrospinal fluid. And so this is how you nourish the brain, because then the cerebrospinal fluid is getting into the parenchyma from the meninges. When there is an inflammatory response ongoing in the gut, then the vessels become impermeable, so there is a downregulation of the PD-1, so no fenestration anymore, and so the dye is not penetrating in the cerebrospinal fluid, and so this way the brain is protecting itself from any possible inflammatory mediators that is coming from the periphery. So it was exactly the opposite of what we were expecting. Then we looked at by single cell RNA sequencing, which were the cells that were responding first to these stimuli that were coming from the periphery. And we found that the vascular endothelial cells were the one that were responding first. And the Wnt signaling pathway was the one that was actually one of the pathway more modulated during this inflammatory response. And the Wnt signaling pathway is important also in the gut. And we know that during an enteropathogenic infection, the Wnt signaling pathway is downregulated, and this allows the PD-1 to go up, and so the increased fenestration of the endothelium. But if we use a mouse in which we don't allow the Wnt signaling pathway, and in particular beta-catenin, to get downregulated, so it remains active all the time, then we are blocking the opening of the gut vascular barrier, and so also a translocation of bacteria across the portal vein. So just by regulating the activation of beta-catenin, we are blocking this rupture of the vascular barrier. So we wondered whether a similar thing was occurring also in the choroid plexus, and so we gave to the mice tamoxifen, which is inducing the elimination of this exon 3, and so the activation of beta-catenin. And we found that also in the choroid plexus, the activation of beta-catenin is leading to the downregulation of PD-1, even in the absence of inflammation. So this is a fantastic model system to study the interaction between the gut and the brain at the level of the choroid plexus without the intervention of the inflammatory response. And since I told you in the beginning that if you have an ongoing inflammatory response in the gut, this is leading to depression, we wonder whether now we could distinguish whether this depression was related to the closure of the choroid plexus vascular barrier, so the interaction between the gut and the brain. And so we use these mice in which we can close the choroid plexus in the absence of inflammation, and we looked at whether the mice display anxiety. And indeed, this is the case. So if you put the mice in this two-chamber cage, then they tend to stay more in the dark chamber if they are depressed. And you can see that indeed, when you are closing the PVB, the choroid plexus vascular barrier, then they tend to stay for longer time within the dark chamber. And they also lose episodic memory. So if you allow them to familiarize with two objects, and then you change an object towards a non-familiar object, they tend to stay with a non-familiar object. By contrast, if you have this impairment, then they lose the ability to remember. So this is not occurring only in mice. So if you take patients that have ongoing inflammation in the periphery, then we could see that there was a change in the choroid plexus volume. So there was a reduction of the choroid plexus in the presence of inflammation in the gut. So also in patients, the permeability of the choroid plexus can be modulated by the inflammatory response in the gut. So to sum up, the gut-brain vascular axis, you have that in the gut under normal conditions, a steady state, the gut vascular barrier does not allow unwanted molecules to penetrate into the circulation. And so the brain does not need to change its permeability, so it can communicate with the rest of the body. However, when you have an inflammatory response ongoing in the gut or a dysbiosis, then this is leading to a change in permeability not only of the epithelium but also of the endothelium. And so there is more translocation of unwanted molecules in the periphery. They get into the brain, and then here the choroid plexus tries to protect the brain by closing down, and so it stops unwanted molecules to penetrate, and so the inflammatory response that can initiate. And this is leading to an anxiety and depression-like behavior. And we now know that this is not only responsible for the anxiety and depression, but also we see this occurring early in life during autism spectrum disorder. So meaning that other neurological disorders can be associated with this change in permeability, which is linked to a change in microbiota composition. So how can we prevent this leaky gut, since this has so many implications in several disorders? And we decided to use postbiotics or hydrogel. So postbiotics, what are they? They are the molecules that are released by microbes that are capable of crossing this inner mucus layer because they are small enough to penetrate. And so these are the mediators of the probiotic activity of the good, let's say, microbiota. And so we compared the activity of postbiotics versus that of the probiotics. And we found that only the postbiotics are capable of protecting the gut from the development of the leakiness. So what we decided to do was to give for 10 days the postbiotics, and then we came with Salmonella typhimurium, which is an enteropathogen capable of disrupting this gut vascular barrier and also all the protection of the gut, including the mucus. You can see here that when you give Salmonella or Salmonella plus the vehicle, then the mucus layer is completely compromised. By contrast, when you pre-treat the mice with the postbiotic, you are preserving the mucus layer, but you also avoiding the upregulation of the PV1, and so the increased permeability of the endothelium. And this is leading to no translocation of Salmonella in the both in the gut and eventually also in the liver. So we are protecting the mice from this leaky gut and eventually also from neurological disorders that are associated with leaky gut. I tell you that when mice are aging, then there is a spontaneous increase of the leaky gut, and this may be responsible for the development of other neurological disorders like Alzheimer's or Parkinson. So then we also use a hydrogel, and the reason being that the structure of this hydrogel resembles quite a lot the mucus layer, so the mucus composition. And so you can see here that when we provide to the mice a high-fat diet, which is also responsible for increasing the permeability of the gut and reducing the mucus layer, that this hydrogel to different concentrations can protect the barrier and also the increase of ZO1, so it's protecting from the increased permeability of the gut and the translocation of bacterial products within the circulation. So this mucus hydrogel is substituting the mucus, and so is protecting the mucus from in the epithelium from unwanted molecules penetrating into the gut. So we have different ways now to intervene via the microbiota with postbiotics, with hydrogels, with even with probiotics in order to protect from the leaky gut. And with this, I wanted to thank my collaborators And in particular, Sara Carloni, Alessandra Silvestri, Juliette Mourier, and Giuseppe Penna, and also Francesca Algeri for the last part. And I thank you all for the attention.
Thank you very much, Maria. Thank you very much, Maria, for your fantastic talk. And it was really interesting. Oh, so I have maybe the first question. Maria, oh, nice to see you live.
Nice to see you too.
Hi.
Nice for inviting me here.
Yeah, no,
this fantastic day of immunology.
It's an honor to have you here. So I was wondering whether you think that in humans there are different sensibilities, different abilities to block the barrier in the brain relating to intestinal inflammation. Do you have any?
Yes. Can you hear me?
Yeah.
Okay. Actually, that is exactly the point. I mean, we think that there are different barriers, and each barrier is actually associated with different degrees of interaction with the rest of the body. So the choroid plexus, that has always been considered as a barrier formed primarily by epithelial cells, is actually, yes, it is a barrier, but it also allows a lot of interactions with the rest of the body. So we think that at this level, that where you want to make sure that when you are exposed to inflammation, this is not really affecting the brain, while the blood-brain barrier is less easier to be manipulated. So in the end, I think this is the major take-home message of this presentation. Yes.
Maria, may I also continue with a question? I thought that also the idea of preventing autism at a very early time point is quite striking. So what about really looking or having some sort of preventive strategy during pregnancy, or would it make sense to make some sort of screening of the gut microbiota to have are there any biomarkers that could be checked whether mothers should be treated with postbiotics to prevent autism in the child?
What we are seeing now is both in environmental models of autism, but also in genetic models of autism, that the mother has a change in microbiota composition, and this leads to a change in the barrier permeability. And then this affects the newborn, because this microbiota is then delivered to the newborn, and then the newborn also has a defect in the intestinal permeability and in the choroid plexus permeability. That's what we know. I mean, I didn't. These are just preliminary data, so they have to be confirmed. But if this is the case, then, as you say, I mean, it would be very important to know what are the differences in microbiota composition of the mothers that might be responsible for this increased permeability. Because then you could, even before birth, make sure that the mother has the correct microbiota and the correct permeability of the barrier, because otherwise this can affect the newborn.
Well, we have questions from the audience, from the online system. Would that protective mechanisms in the brain be also the case in different types of infections, those of non-gut origin, not resulting in leaky gut?
Yeah, this is a very important question, because we still don't know. We do believe that it is possible, because LPS alone, if we give it IV, we also see a closure of the choroid plexus vascular barrier. So suggesting that LPS from any source, that could be from the gut, but also from other infections, might be responsible for the closure. So yes, it is possible.
Yeah, there's another question always from the audience. Does it mean that increased permeability in the gut may induce brain disorders? Yeah. That's what you want to say.
Yeah,
yes, of course.
That's what we want to say, yes.
Yeah.
We still don't have any evidence for neurological neurodegenerative disorders. So for the moment, we have worked primarily on depression and on autism. But we do see, as I was mentioning also in the presentation, that by aging, there is an increased permeability of the gut. And this is probably due to the fact that people that are aging, they tend to eat in a very restrictive way, because they tend to always eat the same type of food, and this is changing the microbiota and is reducing their variability. And so probably this is responsible for an increased permeability. And this, we don't know in the long run. What we have not yet studied is what happens if you have this increased permeability of the gut, which is sustained for a very long period of time, because we did just a chronic colitis, and we see that if we stop colitis, then it takes longer for the barrier to reopen, but then eventually it reopens. But I don't know what happens if you have a continuous increased leakiness of the gut, if at some point the barrier has to open, because then the brain has to interact with the rest of the body, and if this is then inducing the neuroinflammation.
And can I ask you something about the postbiotics? So the good postbiotics, who is producing those? You know, which type of bacteria?
We use the lactobacilli. We know that in children with autism, there are some lactobacilli that seem to have an improvement of the symptoms, like Lactobacillus reuteri. We use the Paracasei because it's a bacterium that we know has anti-inflammatory properties without switching off the immune response. So that's why we use this Lactobacillus, and we actually fermented a fruit oligosaccharide, which is one of the most important HMO, so human milk oligosaccharides. And so it's normally present in the milk from the mothers. But if you have a dysbiosis, also there is a change in this HMO production. So this is why we started with this. So we started with the FOS, the fruit oligosaccharide, and we fermented it. And so we have postbiotics deriving from this fermentation.
Thank you. Do you have other questions?
We have some also comment from the audience that not only the gut itself, but probably a bad situation within the oral, how is it called? Gingivitis, an oral microbiota has an influence on neurological disease. Could you comment on that as well?
Yes, I think this is a very, very important comment. We always focus on the gut microbiota, but we know that the oral microbiota also plays a major role. And also in the oral microbiota, you can have a series of microbes that can cross the oral mucosa, and then also through the veins that are just below the tongue. And this can directly be, if you have an increased permeability there, then the bacteria can go systemic. So this is another possibility, yes. We didn't explore that, but it's a possibility, definitely.
And last question maybe is, what about people who have food allergies and by that reason permanent inflammation within the gut? Is there also a link then to permanent neurological diseases? Have you seen this,
well, we don't know, but also because there are different degrees of opening of the gut. This is also important to point out. That in some cases you have such a high increased permeability that you also have bacterial translocations. In other cases, you just have translocation of some microbial products. So we don't know what happens when you have a mild allergic reactions in which you do have some increased permeability, but maybe not enough to induce so much translocation of bacterial products or bacteria themselves. So we have not studied that, but yes, it could be interesting to study.
Thank you very much, Maria. This was excellent, and thanks to the audience also for a lot of questions. And a few more hellos from the US, from Colombia, and from India. That's a perfect timing to introduce and come to our last issue of the Day of Immunology. And I would like to welcome now Rudolf Valenta, who is the president of this year's 19th IOES conference, which will take place in Vienna, as we said before. And yes, welcome, Rudi. He's here in Vienna, and I think you would give a few highlights and impressions, what people can think of awaiting them in Vienna when they come to our conference.
Well, first of all, let me thank you for allowing me to stand here and to present you, I would say, the Congress in Immunology 2025 that takes place in Vienna from August 17 to 22. Today is a wonderful day in Vienna. thank you, Rita and Ursula, for having organized this wonderful Day of Immunology session here. And I would like to start with a few welcome words by some people who help us to actually organize the meeting that will take in August. And I would like to switch quickly to the words that they have to say, and we will see them in a short video.
The focus of this meeting will be to discuss how we can continue to harness immunology science to develop better therapies across all areas of medicine and develop immunoprevention strategies, for example, to prevent cancer in patients at risk or to enhance healthy aging. We will also be discussing how we can continue to use high-dimensional technology and AI to enhance this drug development effort, and how to develop also targeted therapeutic platform to deliver immunotherapy modality where it is needed. Beyond the scientific exchange, I'm hoping that this meeting will send a powerful statement on the contribution of immunology science to medicine, to really understand the momentum we are in, as it is not the time to scale back investment in science. This is the time to push forward with bold agendas.
The key challenges that will be addressed at IUIS 2025 actually feature all the major topics in immunology. The beauty of a congress this size is that we cover a breadth of topics, from cancer to aging, to infection and vaccines, to allergy, autoimmunity, you name it, we've got it covered. There is absolutely something for everybody.
The program is fantastic. We have organized keynote speakers, two of whom have won the Nobel Prize, so you can meet them. We have symposia, we had a plenary session, and we also have workshops for everybody. So come, because you can learn what's new, what is developing in immunology for our health, and also you will have the possibility to meet all these people to create your own network for your future. So it's good for everybody, for each scientist at every level of the career.
Allergy is the most common immunologically mediated disease in the world. Austrian scientists have been the first to clone allergens and to introduce them into diagnosis and treatment of allergy. Here in Vienna, with the help of European countries, we have developed molecular allergy tests that are available all over the world. And at the moment, we are implementing also molecular immunotherapy strategies for the treatment and prevention of allergies.
We are planning a great social program around the IUS Congress. On Monday evening, we will have a big welcome reception. On Wednesday, we will have an immunology run. And on Thursday, we will have a big conference party in one of the biggest discotheques in Vienna, where also an immunology band will play.
Vienna is a modern city with a fascinating history. Impressive imperial buildings meet modern architecture. And all these famous places are well connected due to an excellent public transport system. On top of art and culture, Vienna is the best cuisine. So in summary, Vienna is livered. Don't miss to waltz with us into the future of immunology for a better health.
Well, dear colleagues, after you have heard these greetings and these words, I would like to come a little bit more into subject. And when going into the subject, there's something that is very important, because we're all immunologists, we like to investigate. And so what I did is I made a little tour by myself on the homepage of the IUS 2025 Congress. And I have to tell you, in fact, it was quite amazing for me because I found a lot of things that I didn't recognize when I made my first tours through it. So let me walk you through it. Well, first of all, today is the perfect day to actually be here and to talk about it because we have a very important time slot. It's the beginning of the late-breaking abstract submission. And actually, you will have the chance to submit your late-breaking abstracts until May 6, 2025. I think that's really an opportunity that you should not miss and you should take. So this is first important message. Now let me come and think a little bit about why you must submit your late-breaking abstract and or register. You can submit a late-breaking abstract, but you can also just come and enjoy immunology. The point now, as of today, I would say the word is true, the cat is out of the bag. What do I mean with the cat is out of the bag? We say in Vienna here, never buy a cat in the bag. You have to look what is inside. And I recommend you to have a look at the exciting scientific program. For this, you click here, the icon program. And then you will come to the many important points and informations. And they are not just empty slots. There is something behind. You can find the keynote speakers, Nobel laureates, you can find the plenary speakers, symposial speakers. You will find the information about each of the symposia. So when you go into the scientific program and you click a certain title of a session, you will find within the session the speakers and also their topics. So the bag is full and you can look into it, what you can find. Another thing that I think is very interesting to many of us, because all we scientists, we wish to publish, and we have actually been successful to organize something very exciting. It's a Meet the Editors session, a panel discussion that will take place on August 20, where actually famous editors from famous journals, Nature, Cell, Press, Science, will actually meet and come together. And of course, this is a very good opportunity not only to meet the editors, but also the editors will be here at the meeting, and you will have eventually the chance to raise the interest of these editors in your work and eventually publish in one of these prestigious journals. Another reason why you must submit your late-breaking abstract and register. This is, we have fantastic activities planned that we call networking events. There is a wonderful welcome reception where we will all be together at the opening of the meeting. There is something that actually people had to convince me about because it's something unique. We will have a congress party, but not at the Viennese Wine Tavern. We will have it in one of the biggest discotheques. It's one of the biggest dancing floors that we have in Europe, the Prater Dome. There are several stages with different types of music, so it's full entertainment, and wir sagen in Österreich, der Zug geht ab, so to say. And there's yet another thing is, and this is important for all those who want to do science, it's important to recover the brain. We learn it today. And how can you recover the brain the best? You do some physical exercise. And for this, we are organizing an immunology run. And there's something very special to this, because this immunology run will actually take place in the famous Vienna Prater at the distance where actually the first human being ran the marathon distance below 2 hours. Now, another very important reason to come to Vienna, and this is something that I recommend cordially to your heart, do not only come to the Congress, but enjoy also at the same time your summer holidays in Vienna. And you can go there and visit the destination Vienna, discover the city, and you will see that Vienna is not only a city, it is more than this. There is a beautiful surrounding. We have, by the way, also childcare capacities during the Congress, so you can come with your whole family. At the same time, visit an exciting meeting and spend the summer holidays in Vienna. So this is something really to be recommended. Vienna is easy to reach. When you go deeper, actually, in this information, you will find hundreds of items and things that you can do in Vienna. I do not only want to mention the beautiful buildings, the museums, the castles, the palaces, the operas, and the parks, also the surrounding and the culture that we have. When you look into this homepage that I present to you here, you can click it and look into it. You will find hundreds of opportunities to spend the time. Now finally, and most important, the meeting will celebrate the birthday of Johannes Strauss, who created the Wiener Walzer, and one of the motives of our Congress is waltzing into the future of immunology for a better health. And I think it's close to our heart of immunologists. What we all want to do is with our work, we want to make the life of people better. And this is actually a central topic in our meeting. So what I can say is, colleagues, Vienna waits for you with all the beauties and a fantastic congress, the best scientific program that you can imagine, but besides a cosmopolitan city with music, arts, hospitality, and everything you can imagine. Please come to us, see the meeting, bring your family, spend your holiday here. We wait for you. Thank you very much.
Thank you very much, Rudi. I'm sure that you could persuade some more people to come to Vienna, and we will be happy to welcome you. Vienna also has a history of great immunologists, actually, and I'm very happy now to have here the president of the Austrian Society of Allergology and Immunology. It's Wilfried Ellmeier. He has also been the head of the scientific board of the conference, but importantly, he's currently the president of our society, and yeah, you will give some new impressions of things of Vienna which not everybody knows.
Okay. Ursula, thanks for the nice word, and also thanks to Rita and Ursula and IOS for giving us the opportunity today also to present the Austrian Society of Allergology and Immunology. And you have heard it already. I think all of you know that Vienna in Austria is famous for classical music, for concerts, for the castle, for horse riding. And for music, Johann Strauss, 200th birthday, as was already said. However, I'm not sure whether you really did know that there's a long and a traditional history of Austria in immunology research. There were important contributions of Austrian physicians in the second half of the 19th and the first half of the 20th century of this emerging new science, which we now know as immunology. Karl Landsteiner discovered the blood groups. Clemens von Piquet, a pediatrician, coined the term allergy for the first time. And there were many, many other scientists, Max von Gruber, Richard Paltau, Rudolf Krauss, Ernst-Peter Pick, Ernst Löwenstein, Michela von Eisler-Terramare, and Anna Mona Spiegel-Adolf, and many others who really did groundbreaking research at the time. However, after two devastating world wars, after severe human tragedy and loss during the Nazi regime, there was also a big loss of intellectual capacity and research and science in Austria. And it took until the second half of the 20th century as Austria could recover. One of the big steps, or one of the first steps, was actually with respect to immunology research, was the founding and establishment of the Institute of Immunology in 1967 by Karl Steffen. And this was the first immunology institute, or institute dedicated to immunological research in the German-speaking countries. And since then, immunology research is thriving in Austria. And today, Austrian immunologists are well connected nationally, at an international level. There are many, many research institutions in Vienna and outside of Vienna. And please don't forget, Austria is a small country with a population of 9.2 million people. So these are really a lot of research institutions which have research foci on immunological and allergological research. In addition, just as a sign of how immunological research is flourishing, there are special research programs and doctoral programs dedicated on a long-term run from 4 to 8 years, just focusing on certain topics of immunological research, on epigenetic regulation of T-cell mediated immunity, on metabolic regulation of the immune system, of JAK-STAT signaling and chromatin landscapes, and also about allergy research funded by the Danube Allergy Research Cluster. In addition, there are special doctoral programs funded by the Austrian Science Fund about securing host immunity. There's one in Graz about immune modulation, respiratory diseases, and also one in Innsbruck about cellular basis of diseases, molecular control of metabolism and inflammation. And these are just a few of ongoing research programs. And these programs just listed here have a total funding of approximately 38 million euros over a period of 4 to 8 years, just indicating that immunology research research is really strongly supported in Vienna and Austria. So immunology is a thriving field in science in Vienna and Austria. And in addition to the programs mentioned here, there are several PhD programs with a focus on immunology. So if you want to do immunological research, Vienna and Austria is the place to do it. So let me tell you a few facts as president of the Austrian Society. Of course, I also would like to mention and briefly introduce to you the hosting organization of the next IOS Congress, briefly introduce ÖGAI, which is the abbreviation for the Austrian Society for Allergology and Immunology, which is the national society of all scientists and physicians who deal with the immune system and its diseases. Members of ÖGAI are active in many basic translational and clinical research areas, from allergy, autoimmune diseases, immunodeficiency, infectious diseases, vaccination, and also tumor diseases, so cancer. So ÖGAI was established in 1971. Currently, it has approximately 600 members and 10 supporting members. And in addition, since 2019, ÖGAI introduced also or established a section of the next generation immunologists in order to also include the next immunologists, the next generation of researchers in that field into the Austrian Society of Immunology and Allergology. The ÖGAI connects science with medicine. It provides a stage for the next generation of young researchers, so we pass on the fire of enthusiasm to the next generation. We help to improve the life of patients with allergies and immune disorders. ÖGAI members contribute to the establishment of guidelines and the improvements of guidelines of how to treat patients. ÖGAI is very well, and its members are very well connected worldwide with the international societies for allergology and immunology. Just a few IUS, EFIS, which is the European Federation of Immunological Societies, the European Academy of Allergy and Clinical Immunology, WHO, FOCIS, and also with the young EFIS. And in addition, ÖGAI members are experienced in organizing big congresses. So ÖGAI members organized already twice the IATSI conference, 2006 and 2016, and also the European Congress of Immunology. And also ÖGAI members initiated smaller meetings, such as the famous Tatra meeting in Slovakia and Czech Republic. ÖGAI, of course, gives also prizes for young scientists for the best papers in the field of immunology and allergology. So it's the Karl Landsteiner Prize, the Clemens von Pequet Prize. We award prizes for the best dissertation. Unfortunately, Fritz Melchers passed away, but he was a generous supporter of ÖGAI and donated always a prize for the best dissertation in the field of immunology. And there are also additional ÖGAI dissertation prizes, and ÖGAI awards also travel awards. In addition, ÖGAI gives also award for lifetime achievements, which is the Karl Landsteiner Medal and the Clemens von Pequet Medal. So I would like to end by saying that the Austrian Society of Allergology and Immunology is looking forward to welcoming you in Vienna in the middle of August. So thanks again.
Thank you very much, Wilfried, and I hope that more and more people get inspired to come in summer. And I just see here in the chat, it was not possible to really answer all questions that were arising. So I think maybe this is another good motivation for you all to come in person to the conference in summer and use the time not only to listen to very interesting talks, but really to interact and discuss with experts and in particular with different colleagues. We come to the end of this Day of Immunology by IOS, but not before I would like to hand over to Rita Cassetti, our upcoming president of IOS, to close with some remarks and thoughts about immunology.
Okay, thank you, Ursula. So I want to wrap up this day with a basic consideration. What you heard today demonstrated very clearly that immunology is a discipline that is important in many different fields, from basic research to clinical studies to global health. So we discovered the immunological mechanism, we explain phenomena, then we develop understanding of diseases. We develop also tools to cure diseases and to have a better health. Now, in this time, we have important challenges. We have challenges that are different in different countries because in the industrialized countries, we have the we are becoming older, that we are becoming overweight, and that this is weakening our immune system. So we have new diseases which we really have to combat and prevent also for ensuring, you know, a healthy aging, let's say. In other countries, unfortunately, war and lack of money is disrupting the life of people, and people have no money, no medicine, no vaccines, and this will increase the frequency of infectious disease, old and new, for kids, for children, and also for adults. The other challenge we really have, and Miriam already mentioned before, is the skepticism about science. We really have to fight skepticism about science. We have to find the right language to be understood by people, but also by politicians, because we have to explain that science means, to support science, to support immunology, means to support the future of this world, to support our life in this world. Can IOS help? Yes, because IOS is here for this reason. You know, we want to join efforts, you know, we want to bring together knowledge from different disciplines, as you heard today. We also want to join efforts between different countries, between different continents, because we want to work together for a global health, but also because all together we have also a stronger global voice. So let's work together for all these things, and come to Vienna for the conference, and I'm very happy that you were here to listen to us. Thank you very much for being here, and so many. We look forward to seeing you in Vienna, and I want to conclude saying again, thank you, Immunology.
Thank you to you all.
And we have here our friends.
Okay. Happy Day of Immunology
Happy
all
day
over
of
the world.
Yes. And see you soon.
See you soon.
See you in Vienna.
Bye.